Hi students, this is Dr. Gayathri, and today we are going to discuss about the renal system and functions. So here are the objectives of today's session. You should have an idea about the structure of the kidneys, the main functions of the kidneys, the basic renal processes that are involved in urine formation. So there are three basic renal processes: the filtration, reabsorption and secretion. And finally, you should have an idea about the assessment of renal functions. So first of all, we'll see the parts of the human urinary system. So the human urinary system consists of two kidneys, two ureters that carry urine from the two kidneys into the urinary bladder. Then we have the urinary bladder where the urine is stored, and then the urethra through which the urine that is there in the urinary bladder comes to the outside. So when we consider the urethra, it is shorter in females and it's longer in males, and also in males the urethra passes through the prostate gland and the penis before it opens to the outside. Now if you look at this picture, you will see these are the parts of the human urinary system. So you can see the two kidneys, the two ureters that brings the urine that is formed in the two kidneys into the urinary bladder, the urinary bladder where the urine is stored, and then the urethra through which the urine comes to the outside. So first we'll see the kidneys. We have to consider the macroscopic structure of the kidneys, that is the gross appearance of the kidneys, as well as the microscopic structure of the kidneys, that is the inner structure, the detailed structure of the kidneys. So first of all, we'll see the gross macroscopic structure of the kidneys. So kidneys are bean-shaped organs. We have two kidneys, the right and the left. And these kidneys are located retroperitoneally, behind the peritoneum, on either sides of the vertebral column, in the upper dorsal part of the abdominal cavity. And each kidney is approximately about 10 centimeters in length, 5 centimeters in breadth, and has got a width of 2.5 or thickness of 2.5 centimeters. And each kidney weighs about 140 grams. Now I have told you there are two kidneys, one on the right side and one on the left side. Now on the right side we have the liver. So because of that, the right kidney is a bit lower than the left kidney due to the presence of the liver on the right side of the abdomen. Now this is a vertical section of the kidney, a cut section, vertical cut section of the kidney. So you can see there are two well demarcated areas, outer pinkish area and this inner area. So this outer area of the kidney is called the outer cortex. So outer cortex is usually reddish in color because of rich blood supply. The inner part of the kidney is called the inner medulla. So the kidney has got an outer cortex and an inner medulla. So in the inner medulla, you can see we have renal pyramids. So these are called renal pyramids. So these renal pyramids, they drain into the renal pelvis. So this structure is called the renal pelvis. So the renal pelvis, it opens into the ureter, right? So this entire structure, this whole thing, this whole area, which includes the renal artery that brings oxygenated blood to the kidney, the renal vein that drains out deoxygenated blood from the kidney, as well as the renal pelvis, which sends urine from the renal pyramids into the ureter. All these three structures together is called the hilus of the kidney. Right? So the kidney has got an outer cortex which is reddish in color due to rich blood flow, and an inner medulla which is pale in color. So the inner medulla consists of renal pyramids. There are about 10 to 15 renal pyramids that terminate into papillae. So these papillae, they form minor calyxes. These minor calyces, they unite to form major calyces, and these major calyces then open into the renal pelvis, and renal pelvis sends urine into the ureter. Now, hilers of the kidney consists of all these parts, that is the ureter, which carries urine from the renal pelvis, then the lymphatics and nerves, the renal artery that brings oxygenated blood into the kidney, and the renal vein that drains out deoxygenated blood from the kidney. So all these structures together is called the hilers of the kidney. So that's about the macroscopic structure of the kidney. Now we will see the microscopic or the detailed inner structure of the kidney. So the functional unit of the kidney is called nephron. Each kidney has about 1 million nephrons. So we have 2 kidneys in total. That means in a normal person there are about 2 million nephrons in both the kidneys. So this is the structure of a nephron. See, this is a kidney. The functional unit of the kidney is called the nephron, and this is the structure of the nephron. So it has got the Bowman's capsule with the glomerulus or the glomerular capillary plexus inside the Bowman's capsule. Then we have the kidney tubules. So first comes the proximal convoluted tubule. Then we have the loop of Henle. So loop of Henle has got the thin descending limb, the U-bend, and the thick ascending limb. Then we have the distal convoluted tubule, which opens into the collecting duct. So this entire structure is known as nephron, and nephron is the functional unit of the kidney. So each kidney has about 1 million nephrons, and each nephron is made up of 3 parts. We have the renal corpuscle. So renal corpuscle consists of the Bowman's capsule and the glomerular capillary plexus, or the glomerulus inside the Bowman's capsule. And where is this renal corpuscle located? That is located in the cortex of the kidney. So if you remember the macroscopic structure of the kidney, I have told you the cortex is bright red in color because it has got a rich blood supply. Why is that? The glomerulus is the capillary plexus or the filtration membrane through which the blood filters. So that means the glomerulus gets a good blood flow, and that is why, since these glomerulus and the Bowman's capsule, since this renal corpuscle is located in the cortex of the kidney, that is the reason why the cortex appears bright red in color, because it has got a rich blood supply. Next, the second part of the nephron is the tubule. So the tubule consists of proximal convoluted tubule, loop of Henle, distal convoluted tubule, and the collecting duct. That is the tubule, distal convoluted tubule. So this tubule is located in both. Part of it is in the cortex, part of it is in the medulla. And then we have the distal convoluted tubule opening into the collecting duct. So collecting duct is also located in the cortex and the medulla. So if you look at this structure, this shows the 3 parts of the nephron. So first we have the renal corpuscle, which consists of the Bowman's capsule and the glomerulus inside the Bowman's capsule. Next we have the kidney tubules, which consist of proximal convoluted tubule, loop of Henle, and the distal convoluted tubule. And then we have the collecting duct. So these are the three parts of the nephron. So nephron is the functional unit of the kidney, and each kidney has about 1 million nephrons. Right, so first we'll see the renal corpuscle. So renal corpuscle consists of the Bowman's capsule and the glomerulus inside the Bowman's capsule. Now this glomerulus or the glomerular capillary plexus is the filtration membrane through which the blood gets filtered into the Bowman's space. So if you look at this structure, you can see this is the filtration membrane. So it has got the glomerular basement membrane. First of all, the epithelial cells of the capillaries, glomerular capillary, then the glomerular capillary basement membrane, and then we have the podocytes. So podocytes have got slits in between. So these are the filtration slits through which the blood gets filtered from across this filtration membrane into the Bowman's space. So the function of the glomerulus or the glomerular capillary plexus therefore is to filter blood. So filtration of blood is the first step in urine formation. And then the Bowman's capsule has got two layers, the parietal layer and the visceral layer. The space in between is called the Bowman's space. So the filtered blood, now through this filtration membrane, the whole blood gets filtered except for two things, the blood cells and the proteins. So when blood is filtered across these glomerulus, the whole blood except the blood cells and the proteins get filtered into the Bowman's space, the space between the two layers in the Bowman's capsule. And this is that glomerulus or the glomerular capillary plexus through which this filtration takes place. And this filtration of blood is the first step in urine formation. Next, we have the renal tubules. So, renal tubule consists of several parts. We have the proximal convoluted tubule, the loop of Henle, which has got a thin descending limb, U-bend, and a thick and thin ascending limb. And then we have the distal convoluted tubule, and finally the last part of the or the third part of the nephron is the collecting duct. So collecting ducts, they pass through the renal medulla and opens into the renal pelvis. So these are the parts of the nephron. So you have to remember there are three main parts in the nephron. We have the renal corpuscle, which consists of the Bowman's capsule and the glomerulus inside the Bowman's capsule. So this is the place where filtration takes place. So the whole blood except the red blood cells and the proteins get filtered in the glomerulus into the Bowman's space. Next we have the renal tubules, the proximal convoluted tubule, loop of Henle, and the distal convoluted tubule. When the filtered blood passes through the renal tubules, certain substances are reabsorbed back into the blood, while certain substances from blood are secreted into the renal tubules. So that means reabsorption and secretion take place in the renal tubules. And then we have the collecting ducts. So finally the urine that is formed enter into the collecting ducts and they drain into the renal pelvis. Right. So when the glomerular filtrate moves along the renal tubules, certain substances like water, glucose and electrolytes get reabsorbed into the blood vessels or the capillaries surrounding the tubules. So this process is called reabsorption. At the same time, when this glomerular filtrate is moving along the renal tubules, certain metabolites and electrolytes that need to be excreted from our body are secreted from the capillaries surrounding the renal tubules into the renal tubules. So this process is called secretion. So you can see there are three main processes in urine formation: filtration, reabsorption, and secretion. So out of these three steps, filtration takes place in the renal corpuscle, right? That is, filtration occurs in the glomerular capillary plexus inside the Bowman's capsule, while reabsorption and secretion take place in the renal tubules. Now if you look at this picture, you will see this is filtration. Now here is the Bowman's capsule, this is the glomerulus, so this is filtration. So you can see filtration occurs in the glomerular capillary plexus. So once the filtrate is formed and it enters into the renal tubules, while the filtrate moves along the renal tubules, certain substances like glucose, water, and electrolytes from the renal tubules are reabsorbed into the blood vessels surrounding the renal tubules. So this process is called reabsorption. While certain other substances like urea and creatinine, which need to be excreted from the body, are secreted from these blood vessels into the renal tubules. So this process is called secretion, right? So these are the three main steps in urine formation: filtration, reabsorption, and secretion. So the final result or the end result of this urine formation process is called excretion of urine. So now we'll see the functions of the kidneys. So the main function of the kidney is to excrete metabolic waste products from the body in the form of urine. Apart from that, the other functions of the kidneys include endocrine function, that is, production of certain hormones like erythropoietin, the active form of vitamin D, and renin. Then it helps to control electrolyte balance and water balance. By controlling the water balance, it also helps to regulate the blood pressure. It helps to maintain the acid-base balance in the body, and also the kidneys help to metabolize drugs and in excretion of drugs after its action. Now, out of all these functions, the basic function of the kidney is to form urine. So, if you remember the three steps in urine formation, these were glomerular filtration, reabsorption, and secretion. So, glomerular filtration plus secretion minus reabsorption is what is excreted as urine, right? So, the kidneys, by forming urine, it helps to remove nitrogenous metabolic waste products that are formed in our body, like urea and creatinine. Apart from that, the kidneys also help to maintain homeostasis. What is homeostasis? The internal environment of the body is called homeostasis. The maintenance of constant conditions in the internal environment of the body is called homeostasis. So what is this internal environment? Now we know in tissues we have cells. Now these cells, surrounding the cells we have a small amount of tissue fluid. So cells, when they are functioning, they need to produce energy. So that means they need to get oxygen. So from where do they get this oxygen from? They get the oxygen from the blood vessels, from the arteries. Oxygen will enter into these tissue spaces or the tissue fluid, and from the tissue fluid, this oxygen enters into the cells. So that means cells, they get oxygen from the surrounding tissue fluid. So during cellular metabolism, carbon dioxide and other metabolic waste products are formed. So these waste products cannot be stored or cannot be retained inside the cells for a long time because they are toxic to the cells. So they need to be removed from the cells. So how do the cells where do the cells remove these metabolic waste products? They remove them from the cells into the tissue fluid surrounding the cells. From there it enters into the veins and ultimately it's removed from the body. So that means the cells are always in constant exchange of substances with its surrounding tissue fluid. So this tissue fluid therefore is what we call the internal environment of the body. So for our body cells to function at its optimum level, the conditions in the internal environment have to be maintained at a constant level. So maintenance of this consistency or maintenance of this constant internal environment of the body is what we call homeostasis. So the kidneys play an important role in maintaining this homeostasis. So kidneys help to maintain the blood volume and thereby it helps to maintain the blood pressure. Kidneys help to maintain the electrolyte composition in the body, and kidneys also help to maintain the acid-base balance or the pH of the internal environment. Another important function of the kidney is to produce hormones. So hormones are produced. So hormones are part of the endocrine system. So that is why hormone production is considered as the endocrine function of the kidney. So there are three main hormones produced by the kidney. One is erythropoietin. So what is erythropoietin? Erythropoietin is the hormone that stimulates the red blood cell formation in the red bone marrow of bones. The second hormone produced by the kidney is the active form of vitamin D, that is known as calcitriol or 1,25-dihydroxycholecalciferol. So why do we need vitamin D? Vitamin D is one of the three hormones that maintain the calcium metabolism in our body. So the function of vitamin D is to increase blood calcium level. So why do we need vitamin D? To increase blood calcium level. So why do we need calcium and vitamin D? For the healthy growth of bones, nerves, for the healthy functioning of the nervous system, muscle contraction, right? So we need calcium for a lot of functions. So to do all these things, we need vitamin D, and the active form of vitamin D is produced in the kidney. The third hormone produced by the kidney is renin. So what is the function of renin? It converts angiotensinogen into angiotensin 1. So that means renin activates RAAS, that is renin angiotensin aldosterone system. So finally the result is to activate aldosterone. So aldosterone is a hormone produced by the adrenal cortex, and its function is to increase reabsorption of sodium and water from the distal convoluted tubules and the collecting ducts. So when it increases reabsorption of sodium and water, what happens is the blood volume gets increased. So when the blood volume is increased, the blood pressure is also increased. So the function of renin is ultimately to activate the renin angiotensin aldosterone system and to increase the blood volume and the blood pressure. Right. So now you have an idea about the structure of the urinary system, the parts of the urinary system. Then you have an idea about the kidney. You have an idea about the macroscopic structure of the kidney, and then the microscopic structure of the kidney, that is about the nephron, and you also have an idea about the basic functions of the kidney. Now we'll see how the renal functions are assessed. So here are the tests: urine analysis, blood analysis, radiography, and renal biopsy. So urine analysis, what is that? That is when you examine the urine for several things. You examine the urine for its physical characteristics, you do certain biochemical tests with the urine, and you examine the urine sample microscopically. So what are the purposes of urine analysis? One thing is for health screening, general health screening, to detect any kidney abnormalities or any metabolic abnormalities in the person. The next thing is, if the patient is suffering from any disorders in the urinary system, to get an idea about what kind of condition the patient is suffering from, you do a urine analysis. Third thing is to monitor patients with diabetes. That is because in diabetes mellitus, one of the long-term chronic complications is the damage to the kidneys. So this is called diabetic nephropathy, and therefore patients with diabetes mellitus should be followed up by doing proper screening for their renal functions, because their kidney functions are gradually getting deteriorated. So because of that, you have to assess them regularly for their kidney functions. Now, what are the precautions when you are doing a urine analysis? You have to avoid strenuous athletic training or heavy physical exercises before the test, because if you do that, sometimes small amount of red cells may be present in your urine, and it may give you a wrong interpretation of your urine analysis. Then you have to do the gross urine examination. That is, you have to examine the urine for its physical characteristics, ideally within 1 hour of urine sample collection. And microscopic examination of urine ideally has to be carried out within 4 hours of urine sample collection. Then your urine analysis results will be accurate most of the time. First of all, we'll see the physical characteristics of urine that are checked. So first thing is urine output. Now the normal urine output of an adult has to be 1 milliliter of urine per kilogram of body weight per hour. So the minimum amount of urine an adult should pass in order to excrete the metabolic waste products from the body is 500 milliliters per day. So that is called the obligatory urine volume. If the person's urine output is less than this, it is not healthy, because then the metabolic waste products that are produced in the body will not be properly excreted. They get stored up in your body, and this buildup of metabolic waste products is very toxic to the person and can have a lot of harmful effects. So that is why the obligatory urine volume is 500 milliliters per 24 hours, and if the person does not excrete this amount of urine, it is toxic. So if the urine output is less than 500 milliliters, if it is less than the obligatory urine volume, the condition is called oliguria. Oliguria can occur due to kidney diseases or when the person is severely dehydrated, such as due to severe vomiting, severe diarrhea, or when the person has lost a lot of blood, maybe due to a road traffic accident, maybe during a surgery, maybe due to a fracture of a main bone in the body, right? So that's oliguria. Then if the urine output is more than 2 liters per day, the condition is called polyuria. So what are the causes for polyuria? One condition is diabetes insipidus. That is when the ADH hormone is not secreted properly, or when the kidneys are unresponsive to your ADH. What happens is water reabsorption in the kidneys does not take place properly. So then most of this water is lost in urine. So that is why the urine volume is increased to more than 2 liters per day. Another condition that can give rise to polyuria is diabetes mellitus. Why is that? Because in diabetes mellitus, the patient's blood glucose level is very high. So when the glucose level is high, it exceeds the renal threshold. So once the glucose level exceeds the renal thresholds, the kidneys are no longer able to retain this excess amount of glucose. So then this excess amount of glucose is excreted in the urine. Now this glucose is an osmotically active particle. So when the glucose is excreted in urine, since it is osmotically active, it draws a lot of water with it. So therefore, in a diabetic patient, along with this glucose, a lot of water is also lost from the body. So that is why in diabetes mellitus, there is polyuria. The person loses more than 2 liters of urine each day. Right, so now we'll see the color of the urine. So the normal color of urine is a pale straw color or amber color. Now there are some abnormal colors in urine. If you look at this picture, you can see these are all abnormal colors in urine. Now what are these abnormalities in the urine color? Urine can become red in color or dark in color due to presence of hemoglobins, red blood cells, due to the effect of certain medications and presence of porphyrins. Then the urine can become black gray color due to presence of melanin in a condition known as melanoma. Then the urine will be bright yellow in color when it contains too much of bilirubin. It will be green in color due to presence of biliverdins or due to the effect of certain drugs. It can become orange in color due to certain drugs or due to presence of urobilinogen. And it will be brown in color due to presence of prophylins or urobilins. So that's about the abnormal colors in urine. While the normal color of urine is pale straw color or the amber color. Right. So now we'll see the odor of urine. So the normal odor of urine. So if there are any urinary tract infections, the urine will be foul smelling. And in diabetic ketoacidosis, since the person loses ketone bodies in urine, the urine has the smell of ketone bodies like acetone and acetoacetic acid. So therefore, in diabetic ketoacidosis, the urine is fruity in smell. Next thing is you check the urine for its transparency. So the normal urine is transparent. That is, if you write some letters on a piece of paper and keep it on the other side of the urine sample, and if you try to read it, you will be able to read those letters. So that means read those letters through the urine sample. So that means the normal urine is transparent. So then what could be the reasons for turbidity in urine or cloudy urine? So cloudy urine could be due to presence of crystals, mucus, vaginal discharge, blood cells, yeast, bacteria. Right? So if you look at this picture, here is a clear urine sample, and this is a cloudy or a turbid urine sample. Next thing we would check for is the specific gravity. So specific gravity is a measure of the concentration of dissolved solutes in urine. So specific gravity therefore reflects the ability of the kidneys to concentrate your urine. So that is called the renal concentrating power. So why do the kidneys need to concentrate the urine? That is to conserve body water. If the body fluid volume is low, then the kidneys should not excrete too much of water, because then the person is going to lose further water. So because of that, it has to reduce the urine output and conserve body water. So to do that, the kidneys will have to concentrate urine. So this is what we call the renal concentrating ability. So what are the causes for altered specific gravity in urine? Now, in diabetes mellitus, what happens is the person loses glucose in urine. So that means the amount of solutes in urine is high. So the specific gravity increases. It will be around 1.035. Now in kidney diseases like glomerulonephritis, it will be less. It will be less than 1.02. And in renal failure, what happens is the kidneys become unable to maintain the normal renal functions. So therefore, the excretion of solutes does not take place properly. So in renal failure, the amount of solutes excreted in urine will be less. So that is why the specific gravity is further reduced in renal failure. It will be 1.008 to 1.01 in renal failure. Now in diabetes insipidus, what happens is the person is losing a lot of water, so there is further drop in the specific gravity, because the person is losing a lot of water, so the solutes that are excreted in urine get diluted. So the specific gravity is further reduced. So that is why in diabetes insipidus, the specific gravity of urine is less than 1.003. So now we'll see the biochemical tests we do in a urine analysis. So first thing is pH. So acidic pH could be due to certain renal diseases, or sometimes it could result from consumption of too much of meat. Alkaline pH could be due to renal diseases like urinary tract infections, renal tubular necrosis, chronic renal failure, or it could be due to consumption of more vegetables and less meat products. Next thing we check for is proteins. So if it is low grade proteinuria, that is proteinuria 1 plus or 2 plus, together with presence of white blood cells in urine, it is suggestive of infections in the urinary tract. If it is high grade proteinuria, that is proteinuria 3 plus or 4 plus with red blood cells, that is suggestive of glomerular damage. So that is proteinuria. And what is the protein that is mostly excreted in urine? In plasma, we have a lot of plasma proteins. So the most abundant one is albumin, and therefore in urine, obviously, the mostly found protein will be albumin. Next thing we look for is the CAS. So cellular CAS with brown pigments and the tubular epithelial cells are suggestive of a condition known as acute tubular necrosis. If you have RBC CAS, it is suggestive of glomerulonephritis. Next, we check for glucose. So presence of glucose in urine is called glycosuria. So what is the cause for glucose in urine? That is diabetes mellitus. Next thing we check is osmolality. So if the osmolality of urine is less than 350 milliosmoles per kilogram of body weight, that is indicative of renal tubular damage. Next is the microscopic evaluation of the urine. So we do the urine analysis for three things. We check for the physical characteristics of urine. Secondly, we do certain biochemical tests. Thirdly, we do the microscopic evaluation of urine. So why is this microscopic evaluation of urine done? It is done to see the red blood cells, white blood cells, mucus, epithelial cells, crystals, casts and microorganisms in the urine sample. So that is microscopic evaluation of urine. So that's about urine analysis. So to get an idea about the kidney functions, the urine analysis alone is not enough. There are certain blood tests we need to do in order to get an idea about the kidney functions. So these are called blood analysis. So here are the tests which we do to get an idea about the renal function. So first thing is blood urea nitrogen. So blood urea nitrogen. What is nitrogen? Nitrogen is the end product of proteins, protein metabolism. So these proteins could be either endogenous proteins inside our body in the form of muscle mass, or it could be exogenous proteins, the proteins that we eat in the form of food. So all of these proteins, when they are metabolized, the end product is called blood urea nitrogen or urea. So this urea needs to be excreted by the kidneys. So the normal urea excretion in the kidneys is 10 to 20 milligrams per deciliter or 2.9 to 8.9 millimoles per liter. So if it is increased than this level, it is suggestive of renal failure. That is, when the kidneys are not functioning properly, the kidneys become unable to excrete this blood urea nitrogen properly from the body. So then the blood urea nitrogen level increases. When the kidneys fail to excrete this urea properly from the kidneys, properly in the form of urine, in the blood, this blood urea level increases. So the blood urea nitrogen level goes up in renal failure. The next thing is serum creatinine. Now creatinine comes from the muscles. So since the muscle mass is constant, the amount of creatinine that is produced daily is also almost constant. So the normal amount of creatinine production in the body is about 60 to 120 or micromoles per liter. 60 to 120 micromoles per liter. So therefore, if there is any increase in the serum creatinine level, in the blood creatinine level, what does it suggest? Now it comes from the muscle mass, so it could be either some extensive muscle breakdown in the body, or it could be the muscle mass is normal but the kidneys are unable to handle the creatinine properly. The kidneys are unable to excrete the creatinine properly, and that is why the blood creatinine level increases. So these are the two reasons for increased serum creatinine level. Next thing is creatinine clearance. Now, creatinine clearance is considered as an indirect measurement of glomerular filtration rate. Now, if you remember the glomerular filtration rate, now I have told you when we did the structure of the kidney, structure of the nephron, I have told you the three main processes in urine formation. These are glomerular filtration, reabsorption and secretion. So this glomerular filtration takes place through the glomerular capillary plexus in the Bowman's capsule. So the rate at which this filtration takes place is called glomerular filtration rate. You cannot measure this glomerular filtration rate directly. So what you do is you assess the creatinine clearance, and since the creatinine comes from the muscle mass, it is constant, it is not reabsorbed nor secreted from the body. The creatinine clearance is almost similar to the glomerular filtration rate. So this is how the glomerular filtration rate is calculated using the creatinine clearance. So the urine creatinine concentration in milligrams per deciliter times the urine volume in milliliters per 24 hours divided by the plasma creatinine concentration in milligrams per deciliter will give you an idea about the creatinine clearance, which is almost similar to glomerular filtration rate. So the normal creatinine clearance is around 110 to 120 milliliters per minute, which is almost similar to GFR, glomerular filtration rate, which is about 125 milliliters per minute or 180 liters per day. Right. So there are certain blood parameters that are suggestive of renal failure. What are they? Complete blood count will show low red blood cells and low hemoglobins. Arterial blood gas analysis will show acidic pH with high bicarbonates, with high hydrogen ions and low bicarbonates. Then sodium will be increased, but sometimes it may vary. Potassium, chlorides, phosphorus, magnesium, all these electrolytes will be elevated. Why? The kidneys are unable to reabsorb these electrolytes, so their blood level will increase. Calcium, it will reduce. Proteins, it will reduce. Why? When the glomerulus is damaged, all the proteins will get excreted in urine. Then plasma osmolality is going to increase. Why? The kidneys are unable to excrete the electrolytes, so all these electrolytes are now retained in blood. So that is why in blood the osmolality is increased. So these are all blood tests that indicate renal failure. Another investigation we do to assess the kidney functions is radiography. So there are several imaging studies that help you to get an idea about the kidney functions or the functions of the urinary system. So the simplest test is called the plain X-ray of KUB, kidney ureter bladder region. So it helps to assess the size and shape of the urinary organs, and also gives you an idea about any cysts, tumors, any kidney displacements or any urinary tract obstructions. So this is a plain X-ray of kidney ureter bladder region. Next investigation is called ultrasound scan of KUB, kidney ureter bladder region. So ultrasound scan is a more detailed assessment of the urinary system compared to a plain X-ray of KUB. So this is how the ultrasound scan is done. This is the probe, and this is how on the monitor you can visualize the structures in the urinary system. Next investigation is called renal arteriogram. So what is renal arteriogram? This is that. So through a catheter you induce a contrast medium into the renal artery. So then when the contrast medium passes through the renal artery and enters into the kidneys, you can see how the contrast spreads along the kidney, along the distribution of the renal artery in the kidney. So that is called renal arteriogram. It gives you an idea about the distribution of the renal artery. Next investigation you do is called intravenous pyelogram. So intravenous pyelogram is also a contrast X-ray. You inject the contrast medium and then you do an X-ray. So it is a contrast X-ray taken to see the structures of the urinary system. Next thing is renal biopsy. So renal biopsy is not indicated in all patients with kidney diseases because it's a complicated procedure. And this is how the renal biopsy is done. It's usually ultrasound guided. So guided by the ultrasound scan, you induce the biopsy needle and take a tissue sample of the kidney and send it for histopathology. So this is how the renal biopsy is carried out. So that comes to an end in today's session. So in today's session we had been discussing about the parts of the urinary system, the gross macroscopic structure of the kidney, the microscopic structure of the kidney, that is about the nephron, the functional unit of the kidney. Next we discussed about the functions of the kidney. We told the basic function of the kidney is to form urine. Apart from that, the kidneys also have other functions like maintenance of homeostasis, maintenance of synthesis of electrolytes, synthesis of certain hormones, right? So, and synthetic functions. Next, we discussed about assessment of renal functions. What tests you do to assess the renal function. So under that, we discussed about urine analysis, the blood investigations which you do in order to assess the renal functions, then the imaging studies that help to diagnose the kidney or the renal conditions in the urinary system, and then the renal biopsy. So with that we end today's session, and thank you very much for listening.